Okay, welcome everybody. Happy spring break eve. So close, man. All right. We have just a couple slides to wrap up chapter 13. We left off with a little bit of a couple of different ways that we might be able to look at effectiveness of substances, antimicrobials, chemical agents, and kind of a good segue into the next chapter when we start thinking about antibiotics. We can do some similar things like we see in this image with some different types of antibiotics or chemical agents that we might think about as maybe being effective. In lab, probably a couple weeks after the break, you'll have an opportunity to do something kind of similar to what we're seeing up here with a handful of different products. Maybe potentially you'll have an opportunity to bring something in from your house if you are questioning maybe a cleaning agent or an essential oil, maybe. Is it effective? Is it not? I think it's kind of interesting. So there are lots of ways that we might be able to test this. And these examples here, we've got like some filter paper discs, little hole punches, if you will, with some paper that's pretty absorbent. You may soak those discs in a cleaning agent or in the image with the real bacteria on there. It looks to me like there's some numbers written on those discs. That makes me think those are likely some antibiotics because we can purchase a handful of different antibiotics with known concentrations impregnated into this paper, and it goes onto the plate of bacteria, and maybe it kills it, maybe it doesn't. All right. So we're looking for susceptibility. Does this agent, whatever it is, does it target these bacteria? Does it kill it? If they're susceptible to that agent, you will see a zone of clearing. And if they are resistant, if that substance doesn't do anything to them, we would expect the bacteria to be growing right up onto that or by that disc. All right. In our diagram right here, everybody's targeted at least a little bit by some of these agents. It doesn't tell us what any of it is, just different antimicrobials. So it could be testing a handful of different cleaning products or other substances, and each one, oh, this in this disc was Clorox bleach or Lysol or something else that you might potentially use in your house, right? Or maybe some mouthwash or other things, all right? And then based off of this zone, so first off, we would take a known amount or just a culture of a specific bacteria and pipette a specific amount, 100 microliters or something like that, a few drops goes onto the plate, and then we spread the bacteria all around to give us what we call a lawn of growth, all right? And as soon as we've got that bacteria spread onto the plate, and we give it a couple of minutes to kind of soak into the media, then we apply the discs that have whatever in there. Incubate it up and see, is there a zone of inhibition or not, all right? So really then we can make some measurements, and then based off of the zone size, we might get some hints as far as how effective that agent may be. Looking at some real stuff next door, here we can see some Pseudomonas and Staphylococcus aureus. So a gram-negative rod and a gram-positive cocci are what we're seeing in both of these examples. Two different types of organisms. And like I said, I feel like these are probably some antibiotics, all right? It also might be a good idea to imagine we're comparing the same things on these plates. So 4 different types of antibiotics tested against a gram-negative, and the same 4 tested against a gram-positive. That can give us some clues as far as how these things work. When we think about antibiotics, there are some that we may think of as a broad spectrum, something that could target multiple types of bacteria, positive and negatives alike. or maybe a more narrow spectrum, kind of targeting specific things. One type, a gram negative or a gram positive, and maybe even more specific with the kinds of negatives or positives. So we could maybe get a little bit of an idea if these were the same things and in the same orientation on these plates, whatever this agent was, if it's the same over here, both, I would think, we would call a broad spectrum targeting each. And then as we look down below, maybe not the case. All four appear to be effective with our staph, but only one with the Pseudomonas. And again, if they were in the same location, the one that's effective against Pseudomonas compared to the one on the staph plate, this zone is much less compared to the Staphylococcus, all right? These zones are humongous compared to what we're seeing with Pseudomonas. Pseudomonas is a bacterium that technically is pretty resistant. And if we had another plate, if we slapped E. coli into the mix, another gram negative rod, and had the same situation, we might see that those four technically were effective with E. coli, just like with the staph, but Pseudomonas had three of them that it was resistant to, all right? There's definitely variation when we start comparing microbes and the effectiveness. With that said, with our staph as an example, we could swab a handful of individuals and test against these agents, and we may see varied zone sizes and maybe some resistance as well, because we're harboring different types of staph on our bodies, some of which might be some drug-resistant forms, and we've never had any issues with it, but you may have them on your body, right? And others maybe have forms that are more susceptible. It truly depends, all right? So these types of tests, we could think about with specific agents, but we also think about when we're talking disease stuff, we figure out what we're dealing with, we cultivate it, we isolate it, we identify it, and then the next question is, how do we kill it? How could we combat it? And an antimicrobial test would give us those details as far as this would be your drug of choice. And like I said, we're going to transition into the next chapter thinking about antibiotics in just a second, all right? So that's the idea with this. This is not the same idea, but a similar one as we're looking at these plates here with these zones. Maybe an agent that we want to look at, like our disinfectant for the bench tops could be an example, all right? So here we've got this disinfectant, some broth, and there's an inactivator, something that's going to neutralize that disinfectant, all right? And then we can basically see what happens, what sorts of growth are we getting in these situations. And these are two different temperatures that we're growing them at to try to figure out what's up, all right? And it lets us see if the disinfectant itself is fine, or if it's potentially got some sort of a contaminant in there as it's spread out, all right? We're not plating bacteria on here and testing the disinfectant to see. We're basically neutralizing it to see, is it full of other things? And that could lend to problems with contamination in the lab, right? Maybe your disinfectant, maybe it's a bad batch, right? And so by inactivating it, it clues us into whether or not that's effective. So another kind of segue into our next chapter, antimicrobial resistance is a huge issue when we think about all sorts of different infectious agents. There are numerous ones that we might classify as multi-drug resistant strains. And some of these things set the stage for some super pathogens, things that we may potentially not have any drugs that really show any sort of ability to target these microbes anymore when they maybe once were. And there's lots of potentials for how some of this stuff may be shared from one microbe to another. We hinted on some of this stuff before when we were looking at some of the genetic stuff with some horizontal gene transfer, where a bacterium may harbor some resistance and it could share that on a plasmid with a bacterium that's completely different from that microbe, giving it some of those potentials. And that's really what we're kind of looking at here. The CRE is a carbapreme, a type of antimicrobial, carbapreme resistant Enterobacteriaceae. So these are gut microbes. Stuff like E. coli fits into that category as of Enterobacteriaceae. And there are handfuls of other ones. All right. So these like our little gram-negative rods. All right. So it's becoming a bigger problem in hospital or nosocomial acquired infections. And some of these situations could be like this duodenoscope that is going in, and potentially these situations, you would hope every device that's used on your body is sterile, but some of these instruments become really pretty difficult to get thoroughly cleansed. And that might be a little bit disgusting, right? You would hope if something like this is being utilized on you, that it is sterile, right? As it's going down and into the duodenum. But there are potentials. And in these hospital situations, it may lend to an opportunity for one individual's harboring some of these pathogens or these resistant forms that now we think of as a pathogenic type that in turn gets deposited into some other individual. And that lends to some of these problems. Okay. Stop that share. Start a new one. Okay, so we thought about chemical mechanisms to deal with things. Now we want to think about these for our own body as we think about different types of antibiotics, antimicrobial mechanisms that hopefully are effective at targeting our respective pathogens. And there are a plethora of different types of drugs, some of which we might think about using in a combination. So we've got a couple of different ways that we may be targeting the microbes with these 2 or 3 different types of drugs that in turn, if one of them in our body is a little resistant, the other drug should still be effective. And so there are approaches that might be utilized with this that we hope help to lend to minimizing potentials of super pathogens. And, you know, it kind of begs the question, how effective is that truly going to be? So initially, our first antibiotic that was discovered and really put into widespread use was penicillin. And we'll go through and talk a little bit about some of this in just a second, but recognized as a potential to target some microbes, and then ramped up into broad, broad use in the very early 1940s. There's some, there's lots of things that they talk about with World War II with, oh, this really helped the Allies win the war. This helped, that helped, whatever it might be. Penicillin is one of them that's right at the top of the list as being a substance that was utilized that truly benefited soldiers, right? As we see some of the propaganda here, penicillin saves soldiers' lives, and lots of things about this, right? One thing I really think is pretty funny as we look at this, or maybe disturbing, whatever, different times, right? Simpler times back then. Here's our soldier in a hospital bed, reading a magazine, smoking a cigarette, just chilling, right? Little bit different now, right? He looks happy, right? Because penicillin saved him. Here we can see another image with children getting some antibiotics. And it, may beg the question here, is this a misuse, over prescription, something like that? Do they truly need it? Or are we actually in the clinic and getting it? I don't know the stories behind all of this stuff, but as I see over prescription, it makes me wonder. Going to the doctor as a little kid because you've got a cold and you leave with some antibiotics, and mom feels really happy that she did something to make you feel better, but it really doesn't do anything because cold's a virus and an antibiotic doesn't do crap to a virus, right? So is it over prescription or misuse as we're looking at this image? I think that's what I'd like to think of it as, all right? So there's all sorts of things that we might think about as having some properties to maybe be effective against specific things. Here we've got some all sorts of different compounds from some plants and fungi that may harbor some antimicrobial therapies. Herbalists in China for forever have used all sorts of compounds that have definitely some effectiveness that, in Western medicine, we think about what are some of these molecules. Could we potentially synthesize these things and maybe modify them? We see lots of stuff, derivatives from penicillin as an example, that once we recognize the molecular compound, if we alter it and tweak it, could we get different types of derivatives that may have other targets that could be effective against? So this kind of makes me think some bioprospecting and looking at what sorts of substances could be held in some of these things that, some of them I'm just like, I have no clue what these things are. Probably some sort of tuber, but I don't know. This looks mushroom-like, and probably maybe these all are a type of that. I don't know, right? This probably tells you all of the details, right? But we can't tell that. There are other things that I have read about with like different medieval things, some writings talking about different types of therapies or techniques that were maybe utilized with recipes, right? They tell you how to make these specific things. And in turn, some researchers trying to replicate that have found that going through this process, these compounds truly are very effective. So it, I think, is very interesting to look at all sorts of different opportunities. And when we think about antibiotics, we're really thinking about fungi or bacteria that are producing these things. So other living organisms, why not think about some other things like maybe some of these plants that also could harbor properties besides the fungi, right? So handfuls of players that we will think about as we move through here. Paul Ehrlich, initially very, all sorts of compounds that he's trying to evaluate for something that could be utilized for the treatment of syphilis, right? And finding an agent with 606 that seemed to have an effect. And I know there's lots of details going through talking about this that, if you are curious about some of the founders of all of these things, there are details about all of them that go through and talk about some of the discoveries. With penicillin, how we're starting really kind of this chapter, Fleming was the microbiologist that ultimately recognized it for its potential, but it was Florey and Chain a decade or so later after the discovery that were actually the scientists that brought it into use, widespread use, all right? So Fleming, he's got, and here we can see some of the Petri dishes on the bench. We talk about, and we've seen the Petri dishes in the lab that are all plastic. Back in the day, these were all glass dishes, and you could clean them and sterilize them and have your media in there and reuse them, right? I imagine something like that, you know, a nightmare for the stockroom today, where we've got hundreds of plates in the lab that's used in a specific week, right? That's a massive amount of glassware that you would have to run through autoclaves and then dishwashers and then re-autoclave it to sterilize it and get all your media in there. That's a huge amount of work, right? Not that plastic waste is a good idea either, but it's hard to say which would be better, right? So a little bit different, all right? So these glass dishes, these glass Petri dishes, kind of loose fitting, all right? So Fleming was studying some Staphylococcus, Staph aureus. He was cultivating this stuff up. He wasn't looking for a fungus that could kill it, all right? He comes back to the lab and the following day sees one of his plates that was on the bench top that had some staph inoculated onto it. The lid was a little bit off and there was some fungus that was growing in there. Maybe it was after the weekend, a little bit of time for the fungus to really get in there and start to grow. I don't know the exact timing of all of this, but this fungus is growing on the plate. And initially, you know, you see something on a plate, and maybe as you're in the lab grabbing some of your plates, you might recognize sometimes these plates are contaminated when you get them. There's a little bit of bacteria already growing on them. If they're old, maybe you might see a little bit of fungus on there. Just depends. And sometimes as they're all packed up in the sleeves, that sets the stage for some issues as they're stored in a fridge and just waiting to get into circulation with the lab. All right. So these plates, this plate that he sees has some fungus on there. And initially his first thought is, well, this plate's messed up. I'm going to have to discard it. But he's a scientist. He looks at it and makes some observations as he sees it. The fungus, as it's growing on the plate, there's no hint of any Staphylococcus growing anywhere near it. But on the other side, far apart from where the fungus is at, the staph is growing. So he starts to wonder, what is it that's in this fungus that's inhibiting my bacterial growth? And he shifts gears the littlest bit to look at this new question. What's going on in this fungus that makes my bacteria not be able to grow? And a substance in penicillium that we call penicillin is being secreted into the media, or the fungus itself, this mold in its environment, would be releasing it out to inhibit things like microbes, like bacteria that may want to also be eating the same food that the fungus might want to be eating. So it's a mechanism for the fungi to ensure it gets a full meal, right? Preventing other stuff from getting in there. Kind of a brilliant strategy. And we're like, wait a minute, we could use that for ourselves, right? And these guys ultimately ramp it up, figuring out how they can get big quantities of it produced from the fungus to in turn extract it and inject it into soldiers or wherever else we start using it. In the very early 1940s, the thought with the discovery of this penicillin really was this kind of magic bullet, the potential to cure anything and everything. Just a quick shot of penicillin and forget about it. You'll be just fine, right? And as we said with the misusage, treating things that it's not effective against sets the stage for other problems. And we see some microbes developing resistance really pretty rapidly against penicillin, all right? Staphylococcus, a couple of years after the widespread use of penicillin, early 40s, by the mid 40s, right toward the end of World War II, we see massive amounts of staph that are resistant to penicillin. It only took a handful of years, all right? Resistance is something that technically is a natural thing as well. Some microbes have capabilities of dealing with these substances, so they can eat that food, right? And through our usage of these agents, that's really more artificial selection. As we start bathing ourselves in these antimicrobial agents, it sets the stage for some that may harbor a little bit of potential to resist that, to really get their populations up and become the dominant type. And they are the ones that are resistant. Kind of crazy. So Waxman, he was one of the first showing all sorts of other culprits that could be effective at producing antimicrobials. All right. So some soil bacteria, the actinomycetes, lots of different types of actinomycetes. This is a kind of bigger umbrella group, and numerous ones fitting into this category that have properties of being able to secrete some agent that targets different types of bacteria or fungus, right? Kind of just depends. And the same with some of the fungi targeting bacteria or some fungi, all right? Just dependent upon what they're producing, all right? So he looked at all sorts of different things and sampled soils and grew up microbes from these soils and then tested these microbes to see, does this type of actinomycetes, does it harbor something? So like cultivating it with some Staph aureus or some E. coli, whatever it might be, to see, does this bacteria kill this other bacteria, right? So one of them, Streptomyces, and we get streptomycin from the Streptomyces. They are a type of actinomycetes. And in the soil, it's a lot of these guys, the Streptomyces and other actinomycetes, but it's a lot of these bacteria that are in the soil that is what makes soil smell like soil. And I think that is really pretty impressive, all right? It wasn't until I took a microbiology class that I learned and understood that, all right? I thought dirt smelled like dirt because it's dirt. That makes sense. Dirt's dirt, right? Don't eat it, right? And, you know, we're in Idaho. I grew up on a farm. I've been in potato cellars. I've smelled this stuff. You smell that earthen odor. It's not just dirt. It's the actinomycetes. It's streptomyces, really, that you're smelling that gives you that kind of odor. After a rain, or you drive past a farmer's field after it's been tilled up, you can kind of smell that smell of soil. That's really what you're smelling, all right? To me, that's a type of bacteria that's okay to smell, all right? You've probably opened up the incubator in the lab and it doesn't smell like freshly tilled soil. It might smell like maybe your back end, right? Because the bacteria we're growing in there, that's where those suckers come from in the first place. E. coli smells like poo because that's where it comes from, right? And in turn, that's really why that smells like that, right? Now it's all coming back together, right? Crazy little microbes. Why do you smell so bad, right? There are so few that actually have some sort of an aroma that might be kind of okay. The plethora of microbes really have some off-putting odors, right? You know that stuff's bad news, right? Okay, so this goes back to kind of thinking our drug resistance and maybe overuse, misuse, whatever it might be, all right? Oftentimes, you know, and I would hope that we're a little wiser now and recognize that you really should take your full course of antibiotics. But my gut says lots of us have been prescribed some antibiotics and you take most of them, but then you feel quite a bit better and you're just like, I'm done. and you stop taking them. That's a potential huge issue to set the stage for problems down the road. All right. So that could be some misuse. All right. So there's all sorts of things we think about. A couple of different ideas in here. In this first bit, we've got all sorts of bacteria, and maybe we're looking at our gut. Maybe we're looking at this CRE stuff, right? Or other things could be some potentials, all right? Maybe Clostridium difficile is a bacterium that we want to think about in this case. Our little blue guys up there, all right? And maybe you've got some other bacterial infection, not anything to do with your intestines, and you're prescribed these antimicrobials that happen to be a broad spectrum drug. And really, these broad spectrums set the stage for lots of issues, all right? Yeast infections can be super common when we think about using broad spectrum antibiotics because of this similar idea. You got a lot of normal flora potentially that this broad spectrum drug winds up targeting. More than just the bad guys, lots of the good guys get targeted, all right? And that's what we're seeing here with these broad spectrums killing off a lot of the ones that we would say, these are our friends, right? We want to keep these guys, right? And here we've got a pathogen in the mix. And this guy could be our Clostridium, right? And maybe held in check by the other good guys. It's there as some normal flora, but it also is a potential pathogen given the right opportunities. Could be a situation like that. So these broad spectrums target the bulk of your good guys, but it happens that the Clostridium, or there are others that would fit in this category as well, that have a bit of resistance against that specific agent. Now, all of a sudden, we've got a massive amount of real estate that's opened up. And Clostridium, they're the only ones that are still around that are like, wow, look at all of this. And they replicate and fill up all of this space. Now you've got massive amounts of the pathogen and hardly none of the good guys, right? And so now we've got a situation that we might think of as a super infection. Clostridium difficile is a nasty one to try to combat after it's actually taken hold in these individuals, all right? They quit whatever antibiotic they were taking. They shift over to something that's going to be targeting it, they hope, but lots of times it's, you know, the effectiveness of that is minimal. Trying to repopulate the gut with good bacteria is becoming more of a common practice. Fecal transplants with these situations to bring back the normal flora. And I think we mentioned a bit of that early on in the semester, a little idea of some of that stuff happening. So this could be an idea that sets the stage with something that's there as a pathogen. We could also think of some misuse where you only take half of the, you know, bottle of antibiotics, half of your prescription, typically 10 days or so is about the course of antibiotics. You take half the course, you maybe do 5 days. And after the first couple of days, those drugs get into your body and you start feeling a bit better. You take them for a couple more days and I'm like, I think I'm recovered. I think I'm good to go. There's still half a bottle left, but I don't need them. And you quit taking them, all right? There might be some of these bacteria that we were really trying to target that don't get killed by these antibiotics. They may harbor a little hint of tolerance to this substance, whatever it might be, and they don't get knocked down by 5 days worth. And in turn, they're able to proliferate. And it's kind of like the blue guys over here, where now all of a sudden we've got these resistant ones that have started to show up. In those situations, now you start feeling like crap again. And you go back to the medicine cabinet and you start eating your antibiotics, right? You've given yourself enough time to get these microbes population back up. You start taking that same antibiotic, and now it's not effective. Then you go back to the doctor. The doctor never cultivated anything. They prescribed something that they hoped was going to work. And now they think, well, maybe I should just prescribe this other agent. And you do the same thing, right? And now you've set the stage maybe for some multi-drug resistant types of antimicrobials. You did it to yourself, right? The bacteria may harbor some potentials. And had you taken the full course, you would maybe wipe their numbers down to the potential that your immune response is competent enough to truly target these. Your normal flora is enough to take over and make it very inhospitable for the small handful that are left around. And you ultimately are better, right? So it truly kind of depends on those situations and how you are maybe eating those antibiotics that sets the stage maybe for problems or not, right? So it also depends really, you know, as you get that course of drugs, depends on how you're getting them and how they are maintained within your system. Oftentimes, you know, you think about going to the doctor and they start asking lots of questions, patient history stuff, trying to figure out different things. And some of these questions might seem like that doesn't really, you know, matter. Why are you asking me this, right? Why do you care how many alcoholic beverages I consume in a week, right? Does that really matter, right? And technically it does. There are lots of things. As we think about someone like trying to get some patient history, maybe a person that is infected with one type of bacteria that doesn't imbibe in alcohol, and another one that's got the same infection but does imbibe in alcohol, the drugs that we would prescribe could be very different. The dosages could be very different. Maybe this person that consumes a lot of alcohol has a huge amount of smooth endoplasmic reticula in their liver to detoxify all of this alcohol that's being consumed. And it sets the stage for their liver to be pretty stinking good at detoxifying lots of stuff. So antibiotics that might be taken, the liver may see it and just say, oh, chew it up and digest it very rapidly, break it down, right? So it doesn't have its effect versus the person that whose liver maybe isn't as proficient at dealing with some of those things, right? Definitely a different situation. That's a little different than this image, but kind of the same idea as we think about how's your body going to deal with it. So this is a route of administration. Oftentimes we think about taking a pill. So the oral administration, it could be a shot like an intramuscular, so IM, or into your veins, intravenous administration. So dependent upon the specifics with that. Some antimicrobials, you know, relying on IV administration, you're going to be in the hospital, right? It's definitely a difficult situation to really deal with some of these infectious agents. Some substances are so potent that IV administration is going to be too difficult on your body, and it gets administered directly into the heart. That's serious. You know you're in bad shape, right? So we can see over the course of time, and this could be days or whatever it is, as it's different administration routes. The intravenous route is the quickest to get the level of antibiotic into your body, in your blood, so it can course through your body. It gets in there considerably faster because you've injected it directly into the veins, right? So we see it spike very rapidly, and then it starts to decline. And over the course of a couple, if we're saying days here, they all kind of reach the same fate. Their levels decline. And this would just be the administration of one thing, one pill, one shot, whatever it might be, all right? Taking in the oral route, a bit of time for it to really get in, to go through the stomach, to make its way into the circulation and start moving through. It truly varies, all right? Oftentimes, we would like to keep the plasma concentration of these drugs at this level right here at our blue or greens as they're peaking, at this level for days, right? We would like it to be that way for about 10 days, high levels, right? The first day you take it, it's going to take 12 hours or so for that pill to really get in there and for that level to get up. But every day, taking it at roughly the same time helps to keep those levels pretty much high throughout the course of your treatment, right? By not taking it properly or missing a day, whatever it might be, that's when we start seeing these levels maybe not at their effective amounts or concentrations within the body, right? So it truly depends on those circumstances. This image talks about expectations of parents and antibiotic usage over the course of two different timeframes from 2000 to 2013. I feel like even at 2000, we should recognize common colds and antibiotics not being effective. It doesn't, I feel like, doesn't matter who you are. We should be clued into some of these details. This image is talking about people that have regular insurance and Medicaid. And I find it a bit irritating with some of this because it seems as if anybody that receives Medicaid feels as if they should get treatment for everything and should just get antibiotics, right? And I think that is not true, right? It also, the impression I get, it makes it seem like these people just don't know what's going on, right? As we see these expectations. And like I said, I truly think that is not accurate, right? But who's to say, right? I don't know where all of this surveying had occurred and all of the details with it, but I personally feel like it's trying to paint a pretty poor picture. So I'm skipping over it. That could be a pack back for somebody, right? There you go. And with that said, pack back closing today, spring break, there isn't a pack back that's assigned. So you don't need to worry about that stuff. You get a week off because you should, right? Okay. I want to just introduce this and then we will stop. All right. So there are boatloads of antibiotics, all right? And we will see as we move through a handful of more of these slides when we get back after the break, some different names. Here's our carbapenems, the ones that we ended with in the last chapter, right? With those resistant ones. Lots of different names, right? I don't want you to get bogged down with all of that stuff, all right? I don't think this course is really where you should be memorizing every stinking antibiotic under the rainbow, because I don't think you need that, all right? But their modes of action, I think, are important, all right? Knowing who fits in here, well, that's maybe for later on. Maybe it's something that will be important. Maybe it's not, all right? And I'm not going to go into huge detail with each individual specific one, all right? So with this, thinking our bacteria, all right, there are a lot of ways that we might be able to target them. And like I said, as we started this, sometimes we might use some drugs in combination with one another to try to target these microbes in a couple of ways, trying to give ourselves the ability to fight them from a couple of different angles, all right? That's sort of the hopes that we don't see super pathogens showing up, all right? So these in bold here are the specific modes of action that these compounds target against our respective bacteria, some of which opens up the potential for thinking about these drugs being more of a broad spectrum, and some others being maybe more limited with that as far as what it might be doing. All right. So the first one, because my pointer was on this to begin with, are the cell wall antimicrobials. So altering the structure as the cells are trying to get in and replicate, it's altering the way that the cell wall is actually able to be produced. So it causes problems within the bacteria and its replication, building cell wall material that makes it unable to complete its life cycle, if you will. So the penicillins that we started with, and a handful of similar ones, are some cell wall inhibitors, all right? Beta lactams, all right? And these guys, pretty stinking good with gram positives, all right? With big, thick cell walls, these are pretty effective with some gram positive bacteria, all right? They can get some gram negatives as well, but they've got, you know, big, thick cell walls and cell wall inhibitors. A lot of these fit into these categories, all right? They're, again, you know, we've mentioned the Enterobacteriaceae, those are negatives, and they definitely can fit some of these, all right? Our plasma membrane, we've got a handful of different potentials within it, where similar, getting in and targeting that and making it so that the microbe can't complete its replication. Protein synthesis, so targeting ribosomes and a couple of different mechanisms, the 30 or 50S subunits of these ribosomes, making it so that they can't go through and produce their proteins. This is important when we think about, you know, some specific drugs maybe having more of an issue on us, causing some adverse reactions. When we think about targeting the ribosomes, bacteria have different ribosomes than eukaryotes, so it's more targeted toward that and not as big of an issue within us. All right. Another one with that would be the metabolic pathways, folic acid synthesis. We don't make our folic acid, we get it from our diet. Drink a glass of orange juice. All right. We get folate and folic acid from our diet, and in turn, microbes, they synthesize it. So if we can block some of these pathways that truly the bacteria are doing on their own, it doesn't cause ill effects in us, and it really targets these guys, right? So there's a handful of different ones that we see here. The folic acid, mycolic acid, if we're thinking like our mycobacteria. So some of the drugs that might be, we think of as targets for tuberculosis and some others could fit within here, right? DNA synthesis or RNA synthesis, so different nucleic acids, this in turn may have more of an impact with us because it could be targeting some of our own processes as well. And oftentimes, like the Clostridium difficile, when we try to, you know, when that shows up and we've got serious issues with it, it's some DNA synthesis inhibitors that are used to target that. You thought you felt like crap with your bacterial infection, and then you start taking these drugs and you feel even worse, all right? And you just want it to do its job. All right, so we'll go through and think some details about this when we get back from the break. And there we go. Farewell. Have a great vacation. If you're traveling, I hope you have safe travels.